Good morning, everyone. Welcome to Grand Rounds. I have the privilege of introducing Dr. KFrank, who is our Grand Round speaker today. She is the chief of the Microbiology Service in the Department of Laboratory Medicine at the Clinical Center at NIH in Bethesda, Maryland. I would like to give you a tiny bit about her background. She received an MD PhD from the University of Pennsylvania. This was followed by clinical pathology residency at Brigham and Women's at Harvard. And then she spent 5 years as a research fellow in immunology with Dr. Fred Alt at the Children's Hospital, Boston. And then she went to the University of Chicago, where she was in the microbiology laboratory from about 2000 until 2012, and then moved to NIH. I'm particularly interested in her work because she has applied a lot of molecular technology to solving hospital epidemiological problems, and I'm excited to have her here today to talk on the spread of resistant gram-negative bacteria. Karen, welcome.
Thank you very much for the introduction, and thank you for the honor of the invitation. It's great to be here this morning. So I have no disclosures relevant to your CME, and the objectives for today are to understand the global relevance of the topic I'm discussing, different strategies that researchers are working to try and combat the spread of the resistance, and I'll specifically be speaking about horizontal transfer of the plasmids that carry the resistance genes. I'm going to start with a case that we had at the NIH. So this was a 16-year-old Kenyan male who presented in Kenya in 2014, nosebleeds, palpitations, headache, fatigue, epigastric pain, and he was found to have a hypocellular bone marrow. He traveled from Kenya to India for therapy. However, he failed the initial therapies they tried to treat his anemia. During this time, he was in and out of the hospital during the year, and he developed a multi-drug resistant E. coli infection, and he was treated with colistin and meropenem. Colistin and meropenem are two of the hardcore drugs we try to save toward the end for therapy. It was determined that he would get a cord blood transplant at the NIH, and he was traveling to the NIH in September of 2015, and he developed fever en route. Upon arrival, he went directly into the ICU. He was in septic shock, bacteremic, and he had a perirectal fluid abscess. This required surgery immediately with a diverting ileostomy for rectal wall necrosis, and he developed respiratory failure and required ventilatory support. His positive blood cultures over the course of his time at the NIH included the multi-drug resistant E. coli, vancomycin-resistant enterococcus, for which we didn't have standard susceptibility testing for the drugs he received, and a resistant Klebsiella pneumoniae. He had a complicated waxing and waning course. He was actually at the NIH for 4 months. Some days at rounds we would hear that he was playing video games in his bed. Another day he was on the ventilator again, so it went back and forth depending on his condition and his infections. And he did receive granulocyte infusions prior to conditioning for a transplant on December 31st. Unfortunately, after the transplant, he developed an invasive mold infection and he died in January. So this is just a snapshot of one of the E. coli isolates showing you the antibiogram and what we had for drugs to give him. So you can see it's almost completely ours with only a couple S's for this particular isolate. This is a snapshot of 3 different multi-drug resistant organisms that he had, the sites of infection, multiple bloodstream infections, and the dates of positive cultures, and the spectrum of antibiotic coverage. So you can see that he had multiple combinations of antibiotics over the 4 months trying to get this under control. And this was the first time in our hospital we had a Klebsiella with an OXA181 carbapenemase. And the point of that is that we don't screen for OXA181. So if it had not been such a striking case that we were paying close attention to, someone coming from India, if it had just been a routine patient, this kind of isolate wouldn't be picked up by our screening. So this case highlights the international arena of healthcare from Kenya to India to the United States and the impact that these resistant bacteria have on the outcome for patients. So what are the large goals in the field? What do we want to do? We want to understand the biology of these resistant organisms, what causes them to be successful as they spread across the globe. We want to slow the spread of carbapenem resistance after we try to understand it, and then we would like to have more effective therapies for these resistant bacteria. Okay, so here's an outline of the talk. I'll describe the carbapenemase enzymes that these bacteria carry and some of the epidemiology. I'll discuss a study of colonization of healthcare workers that we did at the NIH. Discuss the genomics of these plasmids. We've done quite a bit of whole genome sequencing of these bacteria, and specific studies of conjugation or spread of the plasmids between the bacteria. Some of the diagnostic tests available in the study we did to try and work on getting better diagnostic tests. Some drug combination therapy with the robotics facility at NCATS that we work with, and then global relevance. So this is a diagram showing you a carbapenem. This is imipenem, one of the drugs that is in that class, and the carbapenemase enzyme that breaks the imipenem or hydrolyzes it. The most common one in the United States is called Klebsiella pneumonia carbapenemase, or KPC. That's the one you most often hear about. This is a big enough issue that the CDC put a bunch of infections into categories of how much we need to worry about them. And these resistant bacteria went into the urgent category, according to the CDC rules or guidelines that they made. And you can see up to 50% mortality with these bacteria, partly because the patients who get them tend to be sick patients in the ICU who have other comorbidities. They don't tend to affect us walking around most often, but they can be very deadly for these patients. Also, to highlight how important this is, the UN had a meeting in September of this year to discuss these superbugs and discuss things that I'll be talking about, such as can we have a global agreement about controlling the use of antibiotics, particularly for livestock and that sort of thing, that might be important to have these type of agreements. So in terms of a little epidemiology, this is one chart, and I highlight one row of it. It's looking at Klebsiella pneumoniae, and it's over a 10-year period. So showing you that in 2001, if you looked at Klebsiella and you said, how many of these are resistant to just one of the carbapenems, because there's multiple carbapenems in the category, and it was about 39%, with only 2% being resistant to all the carbapenems. Jump ahead 10 years and you see that 69% were resistant to one carbapenem and 10% are resistant to all of the carbapenems. And that's from the MMWR. I tried to find more recent data in terms of epidemiology. This is a 2016 paper. It's a study from Europe, but it is looking at 2013-14, even though it's a 2016 paper because of the delay in the publication. They're only looking at 4 of the carbapenemases from the bacteria that are listed there, but it was 37% of the Klebsiellas and 92% of the E. coli's have these resistance enzymes. So this is a map of the United States from the CDC website showing you what states have reported KPC. So you can see the map is blue, and I'm sure that Idaho and Maine also have KPC, but they just didn't happen to report them. Now, one thing about the terminology, you can see that a lot of the terms are CRE, Carbapenem-resistant Enterobacteriaceae. That term is actually no longer a good term. It should be Carbapenemase-producing organism because it's way beyond the Enterobacteriaceae group. And you can be carbapenem resistant and not have carbapenemases. There's different mechanisms, so you want to be speaking about what the point is of the topic. So this is listing some of the Enterobacteriaceae that I've referred to: Klebsiellas, E. coli's, Enterobacters. The non-Enterobacteriaceae include Pseudomonas species, Acinetobacter, Aeromonas, so these now are carrying the KPC genes. There are greater than 250 carbapenemase enzymes. There are a few that we talk about more often because they're the ones that are more prevalent and or they're the ones that have a high enough activity that they are causing resistance that's clinically relevant. So a combination of those. And I think this is one of the last epidemiology papers, again from 2016, and this is a very big study from the U.S. Department of Health, Department of Defense Health System. So this is the rare group, Walter Reed, and they have a huge sequencing facility, and they gather isolates from across the defense system, not only in the United States, but some of the international hospitals that feed into the United States. They looked at 27,000 isolates, and they pulled out the isolates that had the carbapenemase genes. And the point of my slide is that it had 15 species. So it is not just Klebsiella and E. coli. It is multiple species that they're seeing. Now, what about these other enzymes? So the NDM, the VIMs, and the OXA families are the other ones what we talk about the most often. And what you can see is that the map is not completely blue yet for these enzymes, but the CDC is tracking their prevalence. Okay, one thing that you may have seen in the literature for those who read the lay press who aren't clinical microbiologists that hits to the impact of these bugs. So this was an incident that happened at UCLA Hospital where 2 patients died, but 180 patients were exposed. And it was because the endoscopes that were being used for procedures was contaminated with these bacteria. So a patient had the bug, was given a procedure, and then the next patient had the endoscope used on them even after it had been cleaned. So it was quite a big deal, and what happened was even though the endoscopes were being cleaned per the manufacturer's instructions, the contraption is very tricky to clean, and you couldn't tell if it was really clean. So then what happened for the clinical microlabs in the country is people said, okay, clean or tell me if my endoscope is clean, and we didn't have an SOP to confirm that their endoscope didn't have any bacteria in it. It was quite a hard thing to deal with, and the procedures could be very harsh and actually damage the endoscope. So the FDA got involved and they did modify the mechanism for these endoscopes, but it had quite a big impact on a number of patients. Okay, so this is a diagram where someone's projecting where we might be in the future with these resistant bacteria. So the blue boxes around the ring are showing you causes of death due to things like measles, diarrheal diseases, diabetes, and cancer is a large blue figure there. The purple is projection of where we could be with death caused by antibiotic-resistant bacteria in 2050 if we don't do something about it. So the goal would be that we understand this better, we intervene, and we don't get to this diagram of having that many deaths due to resistant bacteria. Okay, so to put my talk into context, I'm going to be speaking a lot today about the carbapenemase genes and the plasmid transmission of these genes in the bacteria. But in terms of successful spread of a bacteria, why a clone of bacteria can spread from India to the United States, Europe, etc., is not necessarily just because of these carbapenemase genes. So this diagram is just pointing out other virulence factors such as the capsule on the bacteria. I'm not at all speaking today about the capsule on the bacteria, but that's very important in terms of successful clonal spread. Also, to put things into context in terms of what are the big things that people might do to try to combat this spread. One is hygiene practices and facility limitations. So this can be seen as sort of boring by a lot of people, washing your hands, etc., but it's definitely shown to have a high impact, and it's beyond just washing your hands when you do surveillance, how you track it, how you isolate the patients, and the facilities themselves, what kind of cleaning you have in what area, your faucets, etc. Antimicrobial use in animals is believed to have a huge impact. A lot of antibiotics are given for growth promotion of the livestock, and as you'll see at the end of my talk in China, this is believed to be playing a very big role. And then lack of new drugs. As you probably know, many of the pharmaceutical companies are not investing a huge effort in this because when they get an amazing drug, our first thing is to not use it. We want to hold it in reserve for the very sick patient, and they don't get a lot of money, so there are some collaborative efforts between industry and academia or government to try and promote investment in this area of research. Okay, colonization of healthcare workers. So the issue is if you have patients who have this or if you have an outbreak, how much are the healthcare workers involved in the spread of transmission, not just on their hands, but are they colonized? Or do the healthcare workers need to worry that I'm going to get it and I'm going to take it home to my family? I either have a child, I have an elderly person, I have someone who's sick at home, and people get very concerned about this when you have an outbreak. So this is just a snapshot of other papers that have been published in the past. And the point is most of these papers are not carbapenem-resistant bacteria. They're Enterococcus or they're MRSA, etc. I highlighted one that is including CRE. And you can see that it actually had a small percentage, only 1.5% of the healthcare workers in that study were colonized. A fairly big study was done by Adler et al., and they looked not only at healthcare workers, but the family members taking direct care of the patients. And they were looking at gram-negatives, but extended spectrum beta-lactamase, ESBL. And there, they had 3.5% of healthcare workers colonized, and the family members were 9%, so it was higher when they were directly taking care of their family members. So what we did at the NIH, we did a very large study involving 800 people, 400 healthcare workers and 400 controls. And the big deal here was it was a double-blinded study because the concern was if we found a positive nurse in the ICU, would she then have to not be working in the ICU? And what would be the legal ramifications of that consequence? So what we did, it was double-blinded. We did not have their names, and they did not know their own results. So they consented, but then they got a code that only they had, and then it went into the system with a survey so that it could be truly blinded. And the idea was if we had a large positive result, we would then go back and do some other secondary study with more transparency in it. The other thing we did in this study, which was very valuable, was we had a control plate. So this is a perirectal swab, self-collected. It's not actually stool or rectal. And if you're not sampling what you think you're sampling, then you're going to get a negative result. So we put it on a McConkey plate, which is a plate that will show us about gram negatives, and we had to see whether there was some growth at all, that we had some sampling. The skin might have been too clean for the sampling. And most papers do not have that control in them. So the good news, the punchline is we had zero recovery of any carbapenem-resistant bacteria, carbapenemase-positive bacteria in the healthcare workers, and only one in the control sample. The ESBL was about 3-4%, no difference between the groups, which is similar to the literature already, which meant that the healthcare workers were just like everybody else in the community. And this was very, even though it's a negative study, it's a large study, and it's very reassuring for all those people who concerned, or even family members concerned, bringing their patients into our hospital. So we had an outbreak that was highly publicized. So even though we have a very, very low prevalence rate, people were afraid to walk down the street next to our hospital because of the perception from the public. I mean, it really is striking. In terms of people involved in the study, Brooke Decker was the ID fellow who led the consenting with Chris Spalding, the nurse, under Tara Palmore, and then Julie Segre, Anna Lau, John Decker, David Henderson, Sean Conlon, and Nanette Sine were all involved in that study. Okay, genomics. As I mentioned, there was an outbreak at the NIH that was just before I arrived, 2011, 2012. And what happened was a patient came in from New York, was known to be positive, put in isolation, treated, and released. Everything was done per protocol. After the patient was gone, the outbreak started, and interventions were put in place, and it kept spreading, and more interventions were put in place. And they ended up building sort of separate ICU units with walls in the interim. And they ended up with a person 24 hours a day standing in the hallway, and you could not walk down the hallway without being observed washing your hands. Most hospitals do not have the resources to pay someone 24 hours a day to stand in that hallway, but they kept increasing measures to try and get this under control. What you're looking at is a map of the outbreak, trying to track how it spread with traditional epidemiologic approaches, and you can see that you can't track it uniquely. What they did is whole genome sequencing of all these isolates, and the blue dots are showing you single nucleotide changes in the chromosome. And you see going from 1 to 3 to 2, the number 2 has 4 changes in it, which is different than number 3, but the unique positions of the changes are the same. So you would be able to track this uniquely, which you couldn't do previously. This is a bigger map of single nucleotide changes, and what you can see down at the bottom, all the gray boxes look alike. Then you have the, let's see if I can, I don't know how this, so these back boxes look alike, and then these look alike. So you can definitely track that these groups are different than each other, and you will be able to track directionality. So on the right-hand side is the new map. This is how we can track our outbreak. Patient 1 came in. It was spread to 3 unique patients, and then some of those spread it. So what's the point of putting in the effort to do whole genome sequencing on every single isolate for epidemiologic purposes? You can pick up the silent carriers. You can know where to invest effort. So if none of these patients were in the 2nd floor and they're all on the 3rd floor, then you know where to direct your resources to try and get this under control. So that was the outbreak, and since the outbreak, we actually do whole genome sequencing on all isolates that are carbapenemase producers because we're investigating it. This map is showing you the blue was the outbreak. All of the green are KPC positive in the following 2 years, and the purples and the oranges and the reds are the other kinds of enzymes like the oxes and the NDMs. And by looking at this, we're able to tell that we are not getting nosocomial transmission in our hospital. These are being brought into the hospital from outside facilities. And we have some specific stories that we can track with this. So in terms of the sequencing facilities, so the outbreak was clonal, meaning it was one bacteria that was spreading, the whole bacteria was spreading, not the plasmid. We now want to understand the plasmids inside these bacteria. When you do whole genome sequencing with Illumina HiSeq or MiSeq, you get short read sequencing that has to be put together in alignment by the computer and the people guiding that. But you actually don't get the plasmid. So when someone says whole genome sequencing, it means chromosomal sequencing, not plasmid sequencing. To get the plasmids, which fall through the cracks in that analysis, you need a different kind of sequencing. And in this case, it's called PacBio. So it's single molecule reads where you get longer reads. So you combine the longer reads with the shorter reads, and then you can get all the plasmid data. So it takes a bit more work. This is a map of a number of bacteria in our hospital with this kind of sequencing. The rectangles are all bacteria, the circles are all plasmid, and just at a glance you can see that it's a lot of diversity, different numbers of plasmid, different size of plasmid, and if you were looking at the details of the color, you would see what the actual KPC type of gene is on the plasmid, which ones are similar, that sort of thing. And for each case we get in the hospital, we investigate, do we have spread that we need to be worried about? So this is a case of a single patient who had a Klebsiella pneumoniae with a KPC containing plasmid. After the patient had been in our hospital a while, we started picking up an Enterobacter from the patient. So now the patient has 2 bacteria. And the thought was perhaps the plasmid spread from the Klebsiella to the Enterobacter. But by looking at the plasmids, that's not the case. So it was not a case of spread. It was 2 different introductions of the bacteria. And sometimes we don't know where the Enterobacter came from. So if the patient had been in our hospital, but we have no other patient, no other environmental sample, we're not sure if the patient was colonized and then it came out, or whether there was another introduction of the organism. So we can't always get all the details, but we at least do not have a spread that we should be tracking. Okay, this is a different scenario. We had a patient in the hospital who had the outbreak strain plasmid, Klebsiella. And then after the patient is discharged, we're sampling the room, we look at the sink drains, and there's a Citrobacter and an Enterobacter, which have the exact same plasmid, or minor changes, but very similar plasmid. And we believe that this is probably spread of the plasmid in the biofilm of the sink. This particular bacteria has 3 independent plasmids that all contain a KPC gene. So it's as if this bacteria is collecting these, and that's not a bug that you want to have in your hospital. Okay, so there are some patients who survived the outbreak, and they're still patients, so we track them over time, making sure we either isolate them or follow precautions for these patients. And these 2 particular patients, 15 and 16, we sampled over time, and we were able to follow how the plasmids change. So this is one patient, and you can see that the patient has Klebsiella pneumoniae and E. coli, but the Klebsiellas have different ST types. And this is time, so you can see it's 258, 37, 37, then 258 again. So the patient has multiple E. colis and Klebsiellas, and we can track the circles or plasmids, and we can see rearrangements of the plasmids over time. So there's a lot we're beginning to understand about how these plasmids are rearranging in these patients. But this patient is the exception. We don't usually see this amount of positivity and variety. And what we don't understand is what is different about this patient or this patient's isolates compared to the other patients. So this is work we did with the PacBio sequence, but what's very rewarding is that other people then took the work further. So Fred Dite is on the NIH campus, and he and his group are very expert in IS sequences, IS-26 sequences, and they took the same set of data and they analyzed it to an even greater extent. So this is a list of a bunch of mobile genetic elements. So there's different ways that genes can spread around, and I've been speaking a lot about plasmids. What I didn't mention is that the KPC genes are contained on transposons on the plasmids. So that's believed to be playing a role as well, but there's these other ways that genes can spread, and the insertion sequence is one of them. So an insertion sequence is the smallest and most numerous autonomous transposable element. And they're looking at the IS-26 sequences. So they took the same patient, the same set of data, and they were able to track by the scars, if you will, that are left when the IS-26 sequence moves around, the directionality and how, so they're understanding the mechanism of this recombination better than we had understood before, better than the experts who study this had understood it before. So this is rewarding to see the amount of detail we're beginning to understand on plasmid rearrangement. So a summary for the genetic analysis is that we're seeing a lot of diversity. We're trying to track plasmid movement. We are finding it useful in our hospital for infection control. And we're trying to understand the mechanisms better than we had previously with collaborators such as Dr. Dida. Okay, so what about conjugation? So I mentioned those patient 15 and 16, and I mentioned they are not so typical. We have other patients that are colonized, and the only thing we ever get out of them is Klebsiella pneumonia with KPC. We don't get a Citrobacter. We don't get an Enterobacter. We don't get an E. coli. So even though theoretically these plasmids can move, they're not moving in that patient. So what this diagram is showing you is what conjugation is. So you have a pink bacteria that's sensitive. Blue bacteria is resistant with the plasmid. When the plasmid moves from the blue bacteria to the pink bacteria, we now have a new bacteria that is resistant. And we wanted to understand what is controlling this conjugation. Can we understand that better? So what we decided to look at was a number of variables. So the environment of a patient's gut is very different than a biofilm on a pipe. Temperature matters, we thought, or we wanted to investigate that. And the donor bacteria chromosomes and the recipient bacteria chromosomes, as well as their plasmids and any type of mechanism the bacteria had to control gene content. So the bacteria as well want to maintain themselves, if you will. So the experiments were done by the two postdocs in the lab, and what this is showing you is how we set up the experiment. So we took our Outbreak strain, got rid of the plasmid, and then we put in the plasmids that we wanted, sometimes with a GFP tag, but we tried to control all of the variables. Most of the experiments in the field are done with a cloning strain of E. coli. Cloning strain of E. coli loves to get plasmids, so it's a very limited experiment that you get when you talk about the efficiency in that case. The experiments are simple on paper, they're just a ton of work. You have your donor and recipient, you mix them either in a liquid solution or we use a plate and a filter to simulate a biofilm. It's a very rough simulation. You wait a certain period of time and then you plate it looking for how many times do we see the plasmid move? What was our conjugation efficiency? And we can follow it by GFP tag as a confirmation in some cases. So this is a diagram showing you, in this case, the recipient's all the same chromosome. The donor is same or different chromosome, same or different plasmid. So this is a complicated slide, and what I'm showing you is conjugation efficiency on the y-axis. How fast or how easily does the plasmid move around? These are different Klebsiella pneumoniae patient isolates, and this is a 10 log change in results. If it's up here, it means it moves really easily. So that would be like the cloning strain of E. coli. So here the plasma moves a lot. So what you can see is we see a 10-fold, on a log scale, 10 orders of magnitude, not 10-fold, 10 orders of magnitude difference between conjugation efficiency. And if it's an open circle, it means we didn't even detect it moving. If you're familiar with incompatibility groups of plasmids, we looked at the incompatibility group, and that did not correlate. So that was not the answer, just you were trying to put in a plasmid that already had the same incompatibility group. What you'll also notice over here is these are all open circles. So in our lab experiments, the E. coli never took up the plasmid. We know there's E. coli out there that have these plasmids, but there's something that's not being replicated in the in vitro setting. The other thing that we did was look at different kinds of plasmids. So the plasmid called 47E happened to be a plasmid in our 2-year study that we found very frequently. We found it both in the environment and in the patients. And we found it frequently enough, as I said, we weren't seeing nosocomial spread, that it's in the community. It was coming from other centers as well. And it's a very small plasmid. When we did our experiments in broth, we got no transfer of the plasmid at all. Only when we simulated a biofilm did we get conjugation. So this is a plasmid, and it's consistent with what's known about these plasmids, that this was one that probably does need something like a biofilm to move efficiently. So if you're doing these kinds of studies, you need to have your conditions correct for what you're looking for. Now, I've mentioned several times that the outbreak we had was clonal. It was a clonal spread of bacteria. We collaborated with the University of Virginia Medical Center, Amy Mathers, because they had a different kind of outbreak. They had an outbreak of Klebsiella, and in their case, they had multiple species, and they referred to it as a promiscuous plasmid that was moving between the species. So the idea was, could we put these two plasmids from the University of Virginia into our system, and would we see a difference in this plasmid moving more efficiently than the other plasmids? The red dots in this slide are showing you University of Virginia plasmid, and what you would expect if it moved easily would you would see a bunch of dots up here that it moved very easily. Instead, you see some open circles down here as we didn't see it move at all. So again, it's a negative experiment, but what it's telling us is that whatever happened at University of Virginia is not being replicated in the laboratory setting. So it could be that the 10 orders of magnitude that we're looking at don't compare to maybe 100 orders of magnitude if you have selective pressure of antibiotics or an environmental transmission that we're not replicating. So they don't have all the answers of what happened, why did it spread, but it definitely tells us there's more we need to understand here. And we did try and look at the genomics. So we had all of these whole genome sequencing, including the plasmids. So this is a heat map where we tried to look and see, did we see any signals that correlated with our results? The set of genes on the side are all genes that are listed as conjugation genes. And I'll just show you a couple dots. So the blue dot happens to be our outbreak plasmid. It's a plasmid that's very well conserved. It's been reported in Israel and other places in the world with almost no changes. And it has a certain set of known conjugation genes. The red dot is the promiscuous plasmid from University of Virginia. And when we first did this analysis, it was empty. We had no conjugation of genes at all. Like, how can you have no conjugation genes? When we changed our analysis and did a lower percentage identity, we were able to get a little bit of signal down here. So it either means there's more conjugation genes that we haven't identified or don't understand, or you don't maybe need a lot of conjugation genes for this to happen. The gray dot is the environmental plasmid, which has a different set up here. And the green dot is a plasmid from the literature that was again called a high-efficiency conjugation plasmid, an OXA48. And just the point is, it has a different set. So what this tells us is that if someone wants to understand that it's going to be a much bigger bioinformatics analysis and it's going to be a lot of work to piece this out and how much redundancy there is. So the conclusion from this is the conjugation is very contextual, meaning every variable we changed had an impact. So every time we controlled it, we saw a difference. The in vitro studies did not correlate exactly with what actually happened in a hospital, so there's more to understand. The clinical isolates did not accept the plasmids under the conditions that we were testing. And horizontal gene transfer, we are specifically looking at conjugation, but there's other types of horizontal gene transfer, natural conjugation, transduction, and something called membrane vesicle transport. So it could be that some of the things that are happening we're not directly looking at yet, or only a few people are looking at. Okay, diagnostic tests. I realize most of the people in the room are not clinical microbiologists, but I'll spend a couple minutes talking about what we have to worry about when we're looking at this. So we do many, many surveillance cultures at the NIH, even though we're a tiny hospital, about 10,000 per year. And we're still using chromogenic agar. So an agar that has antibiotic in the media and change color depending on species, but we pretty much work up every color on the plate. And someone had mentioned actually last night that there's very little data in the literature about what's available. So this is a review article we published this past year. Robert Bonomo led the group. And these numbers here are telling you how many papers we're looking at that media. 11143. And this was direct swab. So if you look at isolates, you get different data than if you look at a perirectal swab. And because the numbers are so small, the error bars are huge. So what this means is that a lab has to determine, at least with some amount of minimal verification and validation, what they're going to use in their laboratory. And if you look at the larger set of media, many of them are not available in the United States. So there's media available in Europe that we don't have here. So we decided to try and switch to a molecular test. We thought it would be more sensitive. We thought we could implement it, and we thought we would get the resources to do this. This is a list of some of the molecular tests that are out there. Many of them are not FDA approved, or most of them are not FDA approved. A number of them are homebrew assays, so you would have to bring it into your laboratory. Check Direct and Xpert-Carba-R are two of the ones that have gotten further in the field, and one is FDA approved in some cases. So this is a multi-site analysis of the Cepheid Xpert-Carba-R, and it just highlights a few of the limitations. So they had 380 rectal swabs, but they used 250 contrived samples. There aren't enough positives in our patients, fortunately, that we don't have a good spectrum to actually do these studies, so we have to make up the samples to even analyze the assays. They're looking at a subset, only 5 genes here, and they had a discrepancy analysis. So for their assay, it had pretty good sensitivity and specificity as well as positive and negative predictive value, but they have this limitation that they actually had very few true clinical samples that had those specific genes in them. When we looked at it, we looked at Check Direct. We had 300 perirectals. We also had spike samples. Check Direct has a bigger number of enzymes. You can see all these numbers here after these different plasmids. So these are families of enzymes. What I'm showing you here is one graph from our paper. The CT value is the cycle threshold of the PCR. The manufacturer says any positive, so up to 40 cycles you call it positive. What I'm trying to show you here is if you try to say, I'm going to do a cutoff, the red line is my cutoff. Anything below it is positive, anything above it is negative. These red triangles up here are actually negative, but all these gray things are positive, which means if you put that cutoff there, you're missing all these true positives. We were not willing to do that. And if you put your cutoff way up the top, then you have a lot of extra work to sort through this. So the nurse is sitting there waiting to tell you, does my patient need to be isolated? We say, wait, I need 2 more days after my PCR to sort this out. And they're not very happy about that. And it's not really an advantage over the plate, and it costs a lot of money. The other point from our paper here, I just have 2 boxes, and they're showing you limit of detection. And you can just see that sometimes the number matches. It's exactly the same between the plate and the PCR, so the PCR was not more sensitive. However, sometimes the culture is actually more sensitive. Sometimes the PCR is more sensitive. So it is not a blanket statement that the PCR is always better than your plate for sensitivity. So the punchline for R is the sensitivity was excellent. Specificity was not very good with the cutoff that we used. So the positive predictive value was 21%. So we were not willing to go with that. Negative predictive value was great. So if you had the resources, you really can eliminate people and get them out of isolation quickly, but then you have to follow up with the others, and it is expensive. So punchline from the molecular studies is that everybody suffers from having a small sample size and low prevalence. There's a question of what is the true gold standard. That's always an issue of how you're going to compare it and what's your discrepancy analysis. And having multiple targets in the assay, but a limited number of real targets in the patients. There are 2 phenotypic assays that are out there that people have begun to use either a set of or in addition to these molecular assays. One is called the Carba-NP, and it's a colorimetric assay that if the bacteria has the enzyme, chews up the carbapenem, it changes the color of the media. We validated this but never went live because it was pretty tricky for the technologists to actually read the color change. And we had to use 2 different inoculums, and it was a fair amount of work, and you had to have your reagents made fresh. So we didn't go live. We have gone live with this new method called carbapenem inhibition assay, which is pretty easy for the techs. You put your disk of carbapenem in the tube with the bacteria. We do it for 4 hours. Then you take your disk out and you put it on a plate with E. coli. You wait overnight, and then the E. coli grows, and if your disk was unchanged, if the bacteria had no carbapenemase and did not chew up the carbapenemase on the disk, then the disk will kill the E. coli, and you can see this nice circle. So that's a negative result. If the disk drug was chewed up by the bacteria, the E. coli can go right up to the disk. That's a positive. So this is a much easier thing for the text to read. It's like reading a KB plate, and we're finding it easier to use. The reason we're bothering to do this is because our PCR is for KPC and NDM. We are not picking up the other enzymes that we know are out there. We have our plate, but if the plate is positive and the PCR is negative, what is the next step? So we do a lot of sequencing, but that, depending on how you batch it, can take you weeks. So this is something we can do to see if we have a carbapenemase in fairly real time. So how much surveillance is enough? We screen using selective media and the phenotypic assay as well as these 2 CDC PCRs. Many hospitals conduct far less surveillance, and money is a big factor. So what you have to weigh is that we had one outbreak patient that was known outbreak or known positive and was isolated. We still had the outbreak. So it's a really hard decision for administrators to decide what is enough, how far should I go, etc. Okay, just a word on the sequencing technology. So we actually now have an Illumina MiSeq in our clinical microbiology lab, so we can get our results as fast as we want to get them if we want to sequence a batch of clinical isolates. I referred to this PacBio instrument. It's actually a very expensive, very heavy instrument. You actually have to reinforce the floor in some cases to bring this instrument in. This is in a core facility. We send it off and we get results weeks later. I don't know if many of you are familiar, but this is a new technology. It's an Oxford nanopore technology, and we actually have this in the laboratory. It is the size of about a flash drive. This is a computer here, and this is the sequencer. So this is amazing technology, and the way it's working is that the sequence has these, this is a protein pore, and the DNA is threaded through the protein pore, and it interrupts an electrical signal. So you get something like this. It's called a squiggle plot. So depending on how the electrical signal is changed, gives you the read of the sequencing. When we put this sample on, we get the answer in real time. So it goes across the cloud. So even at the NIH, we managed to get this segregated instrument that they let us put stuff on. It goes across the cloud in real time. So we put a bacteria on here, and in 10 minutes, we had a 100% match to an NCBI isolate, no questions, in 10 minutes. Because we had such long reads. So this is very long read sequencing. They've gotten up to 300K nucleotides from a single read. So that's the max. So there's sort of like a competition out there who can get the longest read. Most of your reads are, you can get to 10s and 20s and 50s. You know, it depends on how you optimize your technique. But to get to 40 or 30 is not impossible, as opposed to a 200 read. So 200 versus 10,000. So the people in my lab are very excited, and they're convinced that this is the next wave and that we are going to have these in the clinical microbiology labs because they're so versatile. The limiting factor here is the bioinformaticist to analyze these data. I cannot analyze the data, and I am dependent on the bioinformaticist to tell me what's coming off this sequencer. So it's very exciting. Okay, new drugs or new drug combinations. So this is just 3 slides on a study we did with the NCATS facility, National Center for Advancing Translational Sciences, and they have a robotic instrument or multiple robotic instruments with very tiny volumes for assays. They had an ultra-throughput screen of over 5,000 drugs in combination, trying to find a drug combination to fight these resistant bacteria. So they can do combinations that we cannot do manually. And this was a repurposed screen, meaning the drugs had already, either they already were a drug or they'd been through some level of FDA analysis, so they knew something about the toxicity of them. So these are 2 slides to show you what we got. So we do get a few combinations out of this assay that are worth looking at further. The black bars are showing you growth of bacteria, and these are individual drugs, and some of these you'll recognize these are known drugs. They're just putting them in combination. So the bacteria are growing fine with these doses of regular drug. When we put the 3 drugs together, we were able to see killing. And the red bars are trying to indicate what MIC value. Is it a reasonable amount of drug we could actually give to the patient? And this is a different combination. So each individual drug is not doing anything to the bacteria. In combination, we can get some killing. These are 3 different combinations, which were the best 3 combinations we had in this particular screen. And it was a whole series of bacteria, so it was trying to say more than our one bug could get it. And you can see there's a variety of answers. Some of the combinations worked for all of these. And this is, again, the lines are showing you MIC cutoffs. So even when it went above the MIC cutoff for a sensitive resistant, it was still a dose that you could actually give a patient. So there's a lot out there in the literature about are combination therapies in the laboratory. Can they be translated into using it in a patient? This has been done a lot in cystic fibrosis, and it's been very controversial. I think it's definitely worth exploring this and moving into other phase. So the next phase you would do with any combination you found would obviously be animal studies, etc. But it is a different approach that you cannot do manually in the laboratory. My postdoc cannot do 5,000 combinations in sets of 3 and 4. What we did is we took any combination after they found it and we replicated it in the laboratory. So this work was led by Wei Zhang at NCATS and Peter Williamson of NIID, and Rebecca Weingarten is a postdoc working with me. So finally, global relevance. So I've been speaking mostly about KPC, but NDM is another carbapenemase. And this is a study that was done in New Delhi. And they were looking to see how prevalent is this NDM carbapenemase in New Delhi. And the green and the red dots are showing you either positive in their street water, which is environmental sampling, or drinking water. And the bottom line is they found a lot of positives, unfortunately. So they do have a lot of NDM in India. And there were 14 species of bacteria. It is not just Klebsiella and E. coli. Okay, my very last turn, I'm going to turn away from carbapenemases, which I've been talking about, and talk about colistin. So colistin is a drug that was from the 1950s. It has nephrotoxicity and neurotoxicity, and it had been put on the shelf and not used a lot. It's been brought back, and it's been used, particularly in cases where you don't have anything else to use, such as Carbapenemase-resistant bacteria. These are results that came out of China recently, where they've identified not only colistin resistance, but a colistin resistance gene, MCR1, on a plasmid. After they identified the gene, they sampled by PCR raw meat, animals, and patients, and they found a lot of positivity in China. So this was a very worrisome thing for anyone following this in the field. The other thing that has been found since then is that there's some that have the MCR1 and the NDM in the same bacteria. So it's got the carbapenemase gene and the colistin resistance gene in the same bacteria. On further investigation, these isolates have been found back as far as the 1980s, and in Europe at least to 2004, 2005. So the gene did not just pop out in the past year or so, but it appears to be more prevalent, and it is believed to be connected to the colistin use in the animal livestock. So this is one of the things that we're definitely going to have to explore further and see how fast this spreads. This is, again, a very recent paper, 2016, looking at the MCR1 gene, and this is 35 different countries, either human isolates or livestock. So it is not just in China. This is a map projecting, again, where might we be in 2030, and they're projecting the use of antibiotics in livestock. And you can see there's this huge blue bar for China. The question is, does this need to happen, or can the international community decide that this is not a good idea and we really should control the use of colistin in livestock or antibiotics in livestock? So this doesn't have to be the case. Okay, so I'm going to sum up now with where are the future efforts? Where do people need to invest effort in this field? Hand hygiene in facilities, there's actually quite a bit of work being done on environmental sampling, and we have a very large 2-year project that we're wrapping up now looking at environmental sampling and how do the plasmids differ in the environment. Development of new antibiotics, more robotic throughput may be useful. I mentioned that the capsule was important, very likely, but I did not discuss this research at all. There are people who are working on spread being dependent on good capsule. Horizontal gene transmission, I've mentioned some work, but there's other kinds of horizontal gene transfer that may be important. People are looking at phage therapy, so viruses and bacteria. This is a therapy that's sort of come and gone in waves of popularity, but the idea is, could you design phage that would recognize a bacteria that has a KPC gene in some way or has a certain phenotype and combine 3 or 4 different kinds of phage so that it can't outcompete the phage and give it to the patient in real time to clear their bacteria? We'll have to see, and they're doing it both by screening of sewage, so we actually collaborate with some of these people and they screen lots and lots of phage, or designer phage. People are thinking about what's the best way to design a phage. I mentioned recently just the antibiotic stewardship, or I didn't mention the antibiotic stewardship in humans, so I did not talk about, even within our U.S. hospitals, are we controlling our antibiotic use well enough? Certainly livestock is important. And the last piece is the microbiome. So the microbiome is a huge area of investigation, but people are only beginning to try and understand the microbiome in the human as it relates to these resistant bacteria. I think it's definitely going to play a role given the variety we're seeing in our patients who are long-time colonizers. And then the use of fecal transplants. So fecal transplants have been widely successful for Clostridium difficile. There are some case reports where they have taken a patient and cleared them of the resistant bacteria, perhaps before a procedure, perhaps before a transplant, etc. There are now, I think, 5 clinical trials underway to see if this will hold and if this might be a future strategy for treatment that does not involve finding a new drug. There are a huge number of technologists in the microlab that are critical to all of this analysis of all of these bacteria. Critical leaders in our lab: Gary Foley, Anna Lau, John Decker, Pavel Hills, the bioinformaticist, Maggie Kemp, Jung Ho, Camille, and Rebecca are either senior technologists or postdocs. And almost everything I do is in collaboration with Julie Segre of NHGRI, Tara Palmore of NIAID, and their groups, and Amy Mathers and Anna Shepper were involved in the analysis of the University of Virginia outbreak. Thank you, and I'll take any questions.
That was a great talk, Karen. So now we can open it up to the audience. Dr. Crossan?
Your data would indicate that immune-competent individuals tend not to be colonized with antibiotic-resistant bacteria.
Correct.
Could you comment on the role of the immune system in preventing this, in particular the role of the immune system in plasmid transfer?
So all I can say is I think you're right that it does play a huge role. So you're right that the immunocompetent do not. So you don't need to be afraid walking around the street, even if you walk across the NIH campus. But we don't know very much about it. So there are some people who have gotten pretty far with the role of the immune system in, say, cancer therapies. So they've done some studies with either the gut flora or the skin flora and response to cancer therapies or cancer immunotherapies. People are only starting to look at it with bacteria. So a number of groups are trying to set up animal models, colonized animals, and it's not an easy system to set up. So they're not very far yet, but I think they do need to investigate that. With regard to plasmid transfer, I don't even think people have touched that yet. So there's a lot to do.
Lynn? Dr. Hansen?
That was great. That was a fantastic talk on a very complicated subject. Lots of information. I have kind of a long-winded philosophical question.
Great.
I'm intrigued by your data on the horizontal transfer and the difficulty we're having showing that competent bacteria can pick these elements up. One theory for that would predispose that a mechanism that could explain that would be that these organisms pay too high a fitness price to acquire those resistant elements. Our hospital across the river has a huge problem with oxus, huge problem with oxophenotypes. We've actually sent a couple of our isolates to Patrice in Switzerland to actually characterize, but they're completely benign. We find them on surveillance, they don't cause disease, and Tim Walsh presented data last year at ECCMID showing that oxophenotypes can actually be successfully treated through. And the question exists as whether or not that's a clinical therapeutic response or it's a fitness price that these organisms are taking. Because you have whole genome sequence analysis based on both HiSeq instruments and PacBio, if we have our bioinformaticists aligned to invasive strain genotypes, do you suspect that we will find compensatory mutations or things that might help us understand the fitness price that these organisms may take to acquire some of these resistance genes? Second to that would be if you look at the surveillance data that you presented, clearly a number of patients are carrying as colonizers these organisms yet are not sick.
Yes.
So that would speak to that even further. It's not like it is with C. diff, where we would think that some people could mount a protective antibody response that could allow that colonization. We're unlikely to see patients that have massive antibody response that accounts for their colonization, which would further argue that this is a fitness price that these organisms are paying. Do you have a philosophical comment on any of that?
So the answer is yeah. I've got a paper in my bag that I'm going to read on the airplane on the way back, actually on fitness. So there aren't a lot of people who've yet looked at this with regard to the resistant gram-negatives. So people are starting to look at it. I think there's definitely a role here. My philosophical take is it's going to be a combination of fitness, a combination of surface capsules that play a role, and the gene transfer. So I think the environment is playing a bigger role than we're going to like to admit, that transfer is happening in the environment that doesn't happen on our patients. The issue there is it's a different species. So the environmental microflora is different than clinical isolate. So it may be a good thing that even if it transmits on the sink, it doesn't necessarily infect our patient because it's a different kind of organism. But I think it's going to be a combination of fitness, capsule or other virulence factors, and how the transfer of the plasmid is really happening. We really don't know what happened at University of Virginia yet. So there's a piece missing there. Is it conjugation or is it something else in how it's transferred? And the E. coli. I mean, we only did that one study on the E. coli, but clearly we're not understanding why it's so resistant, even though we understand some of the mechanisms that bacteria have for repelling bacteria from foreign. But I think you're right, and I think fitness is going to be one of those variables.
Other questions? Yes, please.
So you talked about the discovery of the bug in New Delhi. I was in New Delhi at that time. So my question is the discovery of superbugs and these bacteria which are resistant to all these antibiotics, is it an evolutionary change or is it due to some rampant indiscriminate use of antibiotics?
I think there's a lot of data to suggest the use of antibiotics is causing selective pressure. I mean, they're not just for this area, but over many years, if you have certain antibiotics that are used, the bacteria develop resistance patterns. If that antibiotic goes out of favor, the fitness factors play a role in the bacteria come back without the resistance until the pressure is put on again. But there's definitely different use of antibiotics in some of the countries. Even within Europe, different countries have different regulations on how they have it, and their prevalence in their hospitals is pretty different. So I think it's, when you say evolution, when you think about the colistin, the MCR1 gene, it's been around since the 1980s, and we're all of a sudden now seeing an issue. It's partly us paying attention to it, but it's probably selective pressure as well.
Are there any other questions? Yes.
So on the food aspect of this, the animals and whatever, a long time ago, tests were developed so that cows that had penicillin, the milk wouldn't be used, and because it's a big economic loss if you put one cow's milk in a 10,000, 100,000 gallon vat. So there was pressure there to, remedy that. Do you see anything happening on the meat testing or anything like that?
I guess it's not really my field. I don't think there's too much yet. Certainly, globally, I don't think there's too much yet. I think does everyone buy into it? It's important enough, and they need to do something. And what is going to be? Who's going to enforce it? How are we going to get it to happen? I don't think it's moved too far yet. I think certain countries in Europe or the U.S. may move first on that.
Yes, please.
Okay, early in the talk you described the activity of the carbapenemase. If you look across the different plasmids and the different carbapенamases, is there sufficient mechanistic similarity in terms of the activities of these enzymes that it would be practical to design a competitive inhibitor or a suicide reagent that would target those enzymes?
So the answer is there is a lot of variety in their activities. So I mentioned 250 carbapenamases, but we really talk about 4 or 5 of them. The oxas are a category where there's definitely lower activity in some of the oxa categories. Oxa 48 and 181 are the 2 that have been shown in patients and caused outcomes. So the answer is there are definitely changes in activity across the carbapenamases, but that doesn't necessarily mean that someone can't design an inhibitor that hits the most active one. So again, that's not my field, but theoretically they might be able to.
I mean, if the chemical reaction, if the bond is cleaved is always the same one, then it should be theoretically possible to create a intermediate analog that would be transition state analog.
I agree. Are you a chemist?
Many years ago. Yeah.
There's a job for you. All right. Thank you very much for your attention.